One of the greatest unsolved mysteries of regeneration is how the organism know what has been lost after amputation. So for example, if an animal that can regenerate extremities were to lose not the whole arm, but just a tiny little tip of its finger, how does it know that that's the only thing that is missing, such that when regeneration is launched, you don't regenerate a complete arm at the tip of the finger, you actually regenerate the part that is missing.
On the long term, the goal is that our principles can be applied for therapeutics where people who have injury, but also with organ failure, can benefit from regenerative therapies, basically to restore a function that was compromised. It's very well known that depending on how much tissue was lost, the speed of regeneration is different. So we were interested in looking at how does the tissue know how much tissue was lost and how fast it has to regenerate. We used the killifish system. Killifish lives very short lives. In the wild, they don't really need to regenerate, but they do. They have amazing regenerative abilities. What we found is that there are different layers of regulation that will change the speed of growth. What we're proposing is that there is a temporal control, not only which cells and where they are, but for how long these cells are responding makes a difference. So it's a persistence phenotype where we see that the amount of time that it takes for the processes to unfold is different depending on how much tissue is lost. And this persistence mechanism is something that people haven't really studied before.
So if we can in fact modulate the rate and the length of time that a tissue can launch a regenerative response, this would actually help us try to devise therapies that may actually activate and perhaps prolong the regenerative response of tissues that normally would not do so.
Another finding that we observed was the relationship between the skin and how the skin remodels the matrix that is outside of a cell to tell the tissue how much tissue is lost. By remodeling the extracellular space, this recruits new progenitors, and these progenitors read this information from the matrix. So they proliferate and grow as quickly as they should, depending on the information that is deposited there by the skin. We're really opening up new avenues of research when there's this interaction between cells and the extracellular space in the context of memory, how much tissue was lost.
An equally striking finding from Augusto's experiments is that the injury triggered a global response to regeneration, not only in the injured tail, but also in fins that were not injured. And what is striking is that as time passed, that response was completely absent in the uninjured fins, but it was sustained in the injured side, suggesting that injury triggered a global event, but only the injury side allowed that process to be sustained through time. And I think this is actually something that we need to take into consideration, because if you have a degenerative disease in a particular organ, it may very well be that all the other organs are responding to it in a way that we had not really quantified before.
So regenerative therapies are really exciting for the future.
This could allow potentially ways to intervene to restore tissues lost in organisms such as you and me that are incapable of regeneration.